I would like to thank the organizers for inviting me here. It's just wonderful to be in this castle in this far away from Canada land. But I was really pleased last night to watch the football match because Canada's superstar plays on Bayern Munich. You all know this, right? Alphonso Davies. So it was just why he got subbed in, I think a little after half, and it was just great to be here and see him step on the pitch. So just wish I could have been there live. Anyway, I want to talk about task-based fMRI. So I'm really hoping that people will be willing to interrupt and ask questions along the way. There's my slide about nothing to declare. But I want to try to answer these questions. Why do we use tasks with fMRI? I mean, the brain works as networks, and so far quite a bit of the work done on networks and fMRI has been resting state. And so given that the brain works as multiple networks, why would we use tasks? So that's really what I'm talking about today. So let's sort of break this down to starting from what type of information does the scanner provide for us? And when we understand what that information is, what does the task do to that information? And how can we measure the effect of the task? And then finally, what does that tell us? So that sounds no answers, but there will be a slide right at the end with answers to all of those questions, at least my perspective on it. So I won't talk about the physics of it, but fMRI provides numbers that we all analyze that indicate the level of blood flow to where oxygen is needed for neural activity. So it's not measuring any actual information about the neural activity itself, which we just heard about. So if you were hoping to study whole brain neural activity, that's bad news because you're not getting any measurement of neural activity with fMRI. But studying the complex fluctuations of 82 billion neurons, that's highly complex and would be a challenge, as we've seen. But if you're wanting to study robust, straightforward type of brain activity that is influenced by neural activity, the blood oxygen level dependent signal is actually really nice to study. It simplifies this activity of 82 billion neurons into a very simple measurement, which I'll talk about for this entire talk. And so we have one answer, but it's not really that simple because what comes right off the scanner and what we would study with resting state, for example, is hemodynamic response changes. So HDR is hemodynamic response. So what changes in this blood flow, moving oxygen around the brain? Any cognitive event that induces some blood flow change would be sort of mixed together in resting state. Plus breathing, movement, heart rate, arterial dilation, any of those things can also affect the bold signal, plus some noise. So that's basically what's coming right off the scanner. So what can we do with the task? How will that help us? So the task, well, this is supposed to be a depiction of a hallucination. So if there was a hallucination event like that, then there would be the neurons fire in that situation. And if it's detectable by fMRI, that's a big if, but anyway, assuming that it actually is, then there would be this type of hemodynamic response. So notice, let's say that the hallucination was 50 milliseconds long. Then this hemodynamic response actually takes 30 seconds to fully increase and come back to baseline and whatnot. So that's kind of the basic response that we get in the bold signal from some event. But it's not that simple because this is 4 hallucinations that happen. So this is time scale of 30 seconds. So at the bottom here is real time. Hallucination right at the beginning here, and that produces this hemodynamic response. Then another hallucination takes place at 7.5 seconds, and that also produces a hemodynamic response. So that's the second one. Third one here has its own response. Fourth one, of course, has its own response. But that's not the measure that comes from the scanner. What happens is that we have the increase in the bold signal here, peaks, starts going back to baseline, but it's pumped up again by the second hallucination, which peaks, and then just as it's starting to go down again, it gets pumped up again, and then this fourth hallucination will increase it again. So it actually never gets a chance to go back to baseline until here. So we have a very complex type of pattern there. And of course, every single neural mechanism or neural process that induces a bold signal will produce its own little shape like that. And so when you're talking about resting state fMRI, every time someone thinks about what they're supposed to do tomorrow, how loud the scanner is, or any type of neural event that produces a shape, you get a complex shape like that, but in, every single thought and every single process produces different shapes, and it's kind of a very complex pattern of events. So this is just not really math at all, but the way the data could be set up that is retrieved from the scanner. So here we have seconds. So most of the time when we analyze fMRI data, the TR is 2 seconds. So it takes 2 seconds to scan 250,000 voxels, for example. So there may be 250,000 brain areas, and over this 1 to 2 seconds, we'll take a scan of every single one of those and get a number that tells us the bold signal for every voxel for those 2 seconds. Then we have another 2 seconds here, another 2 seconds here, down to the last 2 seconds, time bin, which is 359 to 360 seconds. That makes a matrix with 180 rows because every 2 seconds gets 1 row. Does anyone have any questions? Is that, anyone want to make a comment? So 250,000 variables and 180 rows. So this is 250,000 columns, 180 rows. So if this were resting state data, and all of the cognitive events that take place when a person's resting. So what I have over here is I've just taken that slide of the hallucinations, the 4 hallucinations, turned it so it's vertical, and then just sort of threw it in there. So that would be like one type of thought process and what would happen during rest. So if there were thousands and thousands of thought processes, the thing would be just a complex type of, this whole data set would be difficult to analyze. And that's exactly what we're analyzing when we look at resting state. But it's not, and so for even a hallucination or for any type of cognitive event, it's not just one. Like there are different cell or different areas of the brain that produce different responses even for any type of task. So it's not like you can just, this image that we're so used to seeing of the hemodynamic response like that, that was measured in the visual cortex with a very brief visual display, but nobody really checked the other areas of the brain carefully to see what, and everyone always says, well, there's different shapes, hemodynamic response shapes in different parts of the brain. Yes, because those different parts of the brain are doing different things in response to any type of stimulus. For example, this is, we've done quite a bit we've published quite a bit on working memory, and this is just in preparation right now. But this is actually 5 different brain networks that are involved in working memory. So this task is just remember 4 letters or 6 letters, and then for 4 seconds, and then just see 1 letter and say yes or no whether you saw that. And for that one task, we can measure. Now, I'm not going to get time today to explain exactly how we did this. I have all the slides. If we have time, we can definitely do it. But we have, for example, this early peaking network. It seems to be involved in, if there's a complex set of cognitive processes to take place, this seems to be, when there's been a break and you start, this network activates. And then there's these 3 networks that kind of peak mid-trial, and actually this one as well. But all of them, due to different experimental designs, have different types of processes. Like this one that I call INT is actually internal attention, volitional attention to internal representations. So that is involved in working memory because you have to keep focus on those numbers that are in your some type of holding place there. This red one is a response network that's involved in actually carrying out the response. This is a default mode network here, and this one has to do with focusing on visual features. So we found this set of 5 networks in multiple working memory studies. So we know that for example, for that type of task, and here's the colors of the hemodynamic responses here are on the brain so that you can see which network actually has, on average over the entire network, each of those shapes. So this means that, for example, so if we think about these colors, so actually the light blue, this what we call initiation, this getting online of cognitive processes, has this distribution of these blue areas. And then the default mode network, most people are familiar with that one. So those responses, and I have colors at the top here, I mean, those aren't accurate because I don't even know which XY Z coordinates those actually are. But the colors tell you it could be that that combination of voxels and those brain areas have that response that we saw on this previous slide. And then so the blue might be the default mode network here, so those areas will deactivate. The reds would be the response, and they have their own pattern, right? So within that one task, you have multiple patterns that I just flipped onto here and put down the columns. So again, whatever task it is, there'll be brain areas that have different responses because those networks do different things, different parts of the task they're responsible for. So anyway, what do we have here? Because what does the task do? Produces systematic changes in hemodynamic response against a background of highly complex, non-systematic bold signal changes. So if this working memory task is carried out, people are obviously doing a lot more than that. If you know the percentage of variance in bold signal that presenting the working memory task accounts for, that's about 10%. So 90% of what bold signal you see are from cognitive events or other processes that are not related to the task. So the point of giving the task is just to produce on multiple trials systematic changes in whichever brain networks are involved in that particular task. So I've sort of repeated that here. I think I talked about this already. So what the tasks do, produce systematic changes in hemodynamic response by repeating the same task over multiple trials. Different cognitive operations, even within the same task, produce different hemodynamic response changes in different brain areas / networks. And if the timing or events are known, these networks can be extracted from the data. So if timing of events are known, so this is about timing. I'll talk about that more in just a sec. So how can we measure what the effect the task did? So does anyone want to say anything at this point? Anything come to your mind that you want to talk about? Okay. Nobody, okay. Okay, so we can, if we just have this, just 2x2 table here, in the history of fMRI analysis kind of can be broken down. We have in the rows here the type of analysis. So we can do an analysis that focuses on networks or individual regions. So the traditional GLM is actually carried out as a univariate analysis on every single one of those 25,000 brain areas. But we can also use task timing. So do we have task timing or not? Sorry. And the timing can be from a task, but it can be from EEG signals like we already heard about today, or it could be from eye movements or anything that you have timing about, right? So if you have bold signal, that's great, but do you have timing for some type of event that's gone on during the scan, the session? So if you do, and if you don't, well, normally that's when networks are studied. But I've been going to the connectivity workshop at Whistler for 7 years now, and at the beginning it was a resting state network. And this last time, I think everybody almost mentioned tasks. So it's changing, and I think that's because we have so much publicly available data now, and everyone's sort of done the resting state. Why aren't we looking at tasks? So now all of a sudden people are looking at tasks, but they're still not using the timing. They have the task data and the timing, but they're analyzing it as if it's a resting state scan. So what are we going to do? If we have the timing, how are we going to use it? And usually, that's done as individual reasons in a GLM, like I already mentioned. And rarely, surprisingly to me, because that's all I ever do, is it studied as networks in task-based fMRI. And that has to use timing. So I want to talk about this column. What can we do when we have the timing information? How do we use it? And so that's why I have these two little circles here. We have bold signal on one hand and the task timing on the other hand. So there's two ways we can do this if we want to study whole brain networks. First, we can take the bold signal and we can split it into networks, and then we can look at the way those networks change over all of the TRs for scan series. And then we can correlate that with the task timing information we have, right? And that's the way it's normally done. That's the way ICA works when it's used for network analysis of tasks. So that means or sometimes people use predetermined networks, right? These are the O networks, and these could be computed in advance or just from the scanner and then correlated with task timing. You might conclude this one network is correlated with task timing. Or could do it this way, where you actually look at the overlap between the bold signal and the task timing information and compute the networks based only on that which is shared between the bold signal and the task timing information. That actually turns out to be an overlapping but not the same set of networks. So which one of these is what's the difference between them really, and which is the better one to do? So these networks are optimized to explain the maximum amount of variance in the bold signal, but they're not optimized to be related to the task timing. So if you check post hoc the correlation between the activity in those networks and the timing, you will be guaranteed not to have the brain networks that are optimized for task. But some people don't think you should change the brain networks that come from bold signal, in the sense that that is just sort of like part of the brain, and if you try to take part of it out, that won't be accurate. I don't agree with that at all, and then we do the opposite. We regularly optimize the brain networks to be related to the task timing. And this, like I already said, this basically is what you get from resting state, right? And resting state is hugely complex. You do get reliable networks, but it's hard to interpret exactly what the function of them is. But if we can extract the overlapping variance between task timing and the bold signal, we can optimize the networks to be related to the task timing, and now we're guaranteed to have the best possible networks that we could compute for that particular task, or the EEG signal, or the eye movements, or whatever task timing you have. No thoughts yet? All right, okay. So how do we measure what the task did? Okay, first decision here, when to integrate timing information, before or after, or do we integrate the timing information before or after computing the anatomical computations? So I have made an argument that I think it's optimal to do this after integrating the timing information and the bold signal. Why? Because, just like I said, they're optimized by whatever timing information you have. That actually is very rarely done. Okay, so now we've got two answers. What information is the scanner providing? Talked about that. What does the task do? So I guess I already mentioned that. How can we measure what the task did? We integrate task timing prior to analysis. I think that's optimal. But how? And this is the difficult part. How much time have I used up now? Without you asking any questions, I think I've already used 20 minutes, not bad. Okay, so how do we measure what the impact of the task was on the bold signal? Okay, so we have gone over the bold signal, but what values are used? So you know, the matrix that I showed before, we call it Z in Canada, Z in the US. I'm not sure what you say here. I think you must say Z too. But then we have another matrix now. This is the matrix with timing, which I call G. And so what numbers do we put in there, right? So traditionally, what goes in there is these numbers. And the way it would work for a task analysis where there was only one type of event, like in this case a hallucination, is that we would enter into the software when the hallucination took place, how long we think the hallucination was, and you know there might be short, medium, long, which is actually a really good thing to do. And then that software will compute for us this shape. And that shape is correlated with the actual bold signal in 250,000 brain areas, basically. It's a regression, but same thing if there's only two variables. And then the magnitude of the correlation is actually put onto the brain image, right? So this, I computed this assumed hemodynamic response shape using SPM, and then I just added some random numbers to it, and I'm calling that now actual bold signal. So what we have here is basically the correlation between the model and the actual bold signal, and the higher the correlation, the lighter the color it gets, and under, say, a value that's, you know, a t less than 3 just gets no color, so that's how you get those types of brain images. So that's the traditional general linear model, where we're determining the brain activity by the match of the actual bold signal to the assumed shape, okay? So this, I know, is an assumption that is leading to some misinterpretations, because we now know that there are multiple types of hemodynamic response shapes that are involved in any task. But if we use an assumed hemodynamic response shape to determine our anatomical results of any given task, then we will certainly miss, first of all, it's impossible to get the actual shape that is in the bold signal. If you're close, which you would be in this case, if you use this traditional type, this would be the type of model of the hemodynamic response that would be produced for, when you put in that there's no additional duration, just 0, and this is the traditional shape. That matches almost exactly the initiation network. So if we were to use this model, all we would get is that network, when we already know that there's actually 4 other networks that peak a bit later in different ways. Some of them peak quickly, and some of them take a longer duration. So making this type of assumption will lead to some misinterpretations. Like, for example, I already showed that traditional GLM match. So let's just call this GLM matching, okay? Shape matching, that's what it is. Just trying to match an assumed shape. But we don't have to do that. This is the frustrating thing. We can use a finite impulse response model, which just involves zeros and ones, that tells us the shape. That's how we got these shapes. We don't have to make any assumptions. We could find 5 networks when, with the other way, we'd have 1. And so that a finite impulse response model is slightly difficult to explain, but it's, once you understand it, it's just so straightforward, and it makes everything so much better, I think. So here we have a working memory task, again, 4 letters or 6 letters, 4-second delay, and then people are asked to say yes or no whether or not they saw that letter in the display. So the finite impulse response model works as follows. So here are 4 letters displayed. So this is 2-second duration. Well, actually, the way I put this is not exactly right, but anyway, there's the first scan, 2 seconds. We didn't present anything that one. The next 2 seconds, nothing presented, so all zeros. But on the 3rd scan, we presented 4 letters. And then sometime after that, we presented 1 letter. I have it wrong here because I think, well, it might be right. This is another 2 seconds during that delay, and then this is another 2 seconds, which should start there. So this 2 seconds should be during the delay, so it's not exactly right. I think this R should be down 1 row, but anyway, so then what we're doing here is we're just saying the finite impulse response model says for subject 1, load 4 letters, scan 1. So this is a 1 in this row to estimate the hemodynamic response change on that 1 scan. And then this column estimates on the next scan, this column estimates on the next scan, like that. You just go along for as many scans as you need to cover the amount of time that you want to get the shapes from. So this one we covered about 28 seconds, so maybe that was 14 time bins for 2-second scans, okay? So we just said, let's tell us the hemodynamic response changes that happened during that 30 seconds. Just tell us. We didn't make any assumptions. 1 came out that looked exactly like what we would have put in the model, and all the other ones were just, you look at them, very interesting. It's like it's giving you information. Okay, so this is sort of a graphical depiction of that whole business. So I'll explain it. Don't have to listen if you understood the last one. This is explaining the exact same thing except with some data. So this is the hallucination. So what I did here was I have these 4 hallucinations. I made that nice shape that I already showed you, add some error to it. And then these are just a graphical depiction of the columns of the finite impulse response model. These are zeros and ones. So this one has 1 on the very first scan when the hallucination took place, and then zeros everywhere else until we get to the 2nd scan. There's another 1. Sorry. Till we get to the 2nd hallucination, put a 1 in there. 3rd hallucination, 1 there. 4th hallucination, 1 there. And then this is the same pattern, but it's on the 2nd scan, and this is 3rd, and this is all the way up to 10. So what we're doing is we're asking the analysis to tell us what were the hemodynamic response changes that happened in 10 scans, that's 20 seconds, after the start of the task, okay? So actually, I literally did this in SPSS. So I took this as a dependent variable, and then I took these 10 independent variables, and there was 30 rows in the data, did a multiple regression, dependent variable here, 10 independent variables, and these are the beta weights that came from that regression. And lo and behold, believe it or not, they produce a hemodynamic response shape. It's a little bit tricky to really understand how it does that. It's because every one of these columns of zeros and ones explains the variance over and above all of the other columns. And those of you that are familiar with multiple regression will understand that a beta only indexes variance over and above all of the other ones. And that's why we can actually get this really precise type of hemodynamic response shape. And so we can either do the suboptimal shape matching to an assumed shape, or we can do the optimal retrieval of shapes from we can retrieve the shapes of the task-induced hemodynamic response changes. This is obviously optimal because the anatomical patterns, which is of course what we're after, but they won't be restricted to only matches to the shape, but they will be attached to it's using principal component analysis. So the hemodynamic response changes have to do with component scores, and the anatomical depiction have to do with the component loading. So there's just two sides of the same coin. And so we actually see the anatomical pattern, but we also get the hemodynamic response shapes, which are very, very interesting pieces of information that are completely missed when we just put those shapes into a model and look at the beta weight. So then all we get is the picture. Why not get the picture, all of the networks, and look at the hemodynamic responses that are associated with each network? Okay, and another problem that we have with the matching method is that we can get confounded networks, very highly confounded networks. For example, we can have a patient hallucinate in the scanner, but if they don't tell us when they're hallucinating, there is no really good way to know that they did. So what do? Squeeze a ball, press a button. Guess what? It's perfectly correlated with the actual hallucination. Now what are you going to do? Right? So you get the bold signal, you get the timing. It's like, yes, we know when the hallucinations were. Well, okay, so they're hearing a voice. Yes. Anything else? They were pressing a button. What? Pressing a button and hearing a voice? What are we going to do now? Okay, so I mean, it kind of depends on how long the hallucinations are, or whether they press, how they press the beginning and end. But it could be that the button pressing and the hallucination produce the exact same assumed model, right? So when you do that, if you have the button press and the hallucination, now you're going to get an image, and the betas will match in the response regions and in whatever regions are actually involved in hallucination. So maybe the superior temporal gyrus, and then this is, sensory motor regions, right? So all of those regions correlate that signal. And if you actually ask the hemodynamic responses to emerge from the data, they're not exactly the same. It's just that they were so similar that we couldn't tell the difference because they both matched. Because, the hallucination missed a bit, the response missed a bit, but on average, you couldn't tell the difference because we never looked at the hemodynamic responses. All we looked was at the brain image, and if you were a bit sloppy, you'd just say, "Here's a hallucinations network," right? So, how do we solve this problem? Okay, so this is basically showing this again, showing this is the response. Let's see here. Traditional GLM method may miss light blue while differencing out red from green. So I'll talk about it a little bit more. So when we use the assumed shape model, what people often do is have, say, one task. It's impossible to do, but you could have one with, say for working memory. Okay, we'll go back to that. Okay. You can have one working memory task that involves remembering the letters and responding, another one that only involves responding but not remembering the letters, okay? So this is the response here, the response network, and this green one is the remembering the letters network, then paying attention to internal representation. So if we were to have a perfect match to this on a different task, we could difference out the betas and get rid of this network, right? And so then we'd be left with the working memory network. Problem is that you never get an exact match, so it doesn't exactly work. And so things get confusing quickly. But we can still do it this way, not make any assumptions, and actually we can do a dimensional analysis. So let's see, dimension reduction allows retrieval of these networks. So if we, doing the model matching method is sort of like a flat picture, right? The last one we saw, the superior temporal gyrus network and the response network are on one image. But using a dimension analysis, you can allow the networks that are producing that image to separate out. And that's how we get the separate networks and the hemodynamic response shapes associated with each network. So this is just sort of a depiction that this complex shape, we might get this one for the superior temporal gyrus, but a different hemodynamic response for the response network, which is what we want. That's what we're trying to find, I think. Okay, so anybody want to have any? There's quite a bit I've already said. No ideas? No comments, no questions. One question.
One question. Would you think that you would have to optimize the hemodynamic response function on an individual basis, or
well,
I mean, might be different from person to person.
Well, okay, so I've been arguing for the past 15 minutes that we shouldn't use the assumed shape, right? So are you saying, are you saying that I should not only use that shape, but try to make it specific to each individual? So I would say, why would I even try to do that? Because all I have to do is use a finite impulse response model for every person, and I will get that individual's hemodynamic response shape.
Why are you interested in the hemodynamic response shape in that case?
In the case of?
If you're saying that it's different between different people in different actions, why would you even be interested in plotting it?
That's a great question. I have the answer right here. So let's talk more about the hemodynamic response shape. Why am I interested in the hemodynamic response shape? It's because the anatomy is not enough for me. Maybe it's enough for some of you, but I'm trained in psychology. I want to understand the function of the brain network. And to understand the function of the network, I need to look at how it responds to task conditions. That's the reason. How it responds to the conditions that you built into the task. So we, who are trained in psychology, we don't think of the task as sort of something that's, I don't know what task to give, just do resting state. That's what some people think, right? But those of us that are trained in psychology, from the time we were trained, we thought brain imaging was going to be about tasks. What happened? You know? So now it's 20 years after sort of a resting state took over, 15, we're finally getting back to tasks. And the reason is because we want to understand what these networks, the function of these networks are, for example. And that's sort of what the rest of my time will be spent on, unless people want to talk about something else. But so here is, now this is analysis of this. 4 letters versus 6 letters, 4-second delay, or 0-second delay, or no delay. And then we have 1 letter, say yes or whether or not we saw that letter, okay? So bear with me here, we're getting into psychology a little bit. We were able to retrieve through a dimension analysis, so what we did was we constrained the bold signal to that which is predictable from task timing, 10% around. So we take out 90%, okay? Restrict it to 10% of the data that is predictable from task time. Then we do a dimensional analysis on that, and it turned out there was 4 networks that we retrieved for this study, which is published in Cortex. And so, is the anatomy, so look at that. We can tell that's the response network. There's left lateralized sensory motor areas there, okay? But is that good enough? For some people, I'm just like, are you sure? And actually it turns out that it is important to check the hemodynamic response shape. So we have predictions about what that hemodynamic response shape should look like. One of them is that this task has either 4 seconds delay or 0 delay. And then people respond when they see that letter. So there has to be 2 peaks that are 4 seconds apart in the response network activity. Because either they see that for 4 seconds, respond right away, or they see this for seconds, wait 4 seconds, and then respond, okay? And there is exactly 4 seconds between those 2 peaks. Additionally, by the time the response takes place, there doesn't have to be any load dependence. I'm not saying there could never be, but there shouldn't be really, because you've got your answer and you just respond, yes or no. And so there is this is 4 letters versus 6 letters, and there's really no difference between 4 letters and 6 letters for that response network, okay? So now we go around to all these other ones, and now we get some more information about what they might be doing. Default mode network is fairly easy. I mean, it's usually load dependent, so this is 4 letters, and this is 6 letters for when there's a 4-second delay, okay? So we can see that the default mode is extended out when there's a 4-second delay, and it's these are negative component loadings, so these are actually showing us that there's a decrease, which is almost every task has a decrease of the default mode network. So we have something that starts early, slowly peaks, takes a little longer when there's a delay, and it's a little bit shorter, and it's load dependent. It's a perfect type of pattern for a default mode network. What about this one? And this is that one that I talked about before that kind of peaks earliest. This peaks at 8 seconds. So this one is interesting because it takes a little bit later to start, and then you have a huge difference here between 4-second delay with a high load and no delay with a high load. Okay? Compared to the default mode, here's 4 seconds delay high load, no delay high load. Not really that much difference, huge difference here. So obviously this is very much involved in maintaining the information and more activity when there's more to maintain. So when we have all that information, now we're getting closer to having a functional interpretation of what those networks are. So here's sort of what we're doing here. Integrate task timing in advance. Represent model timing for model instead of an assumed model. Separate brain networks, multiple dimensions. And so, that's kind of the basic principles of this third one. Oh, these are, okay. So, I think this is pretty much saying the same thing, so I'll just go over it again. If you have any questions that it brings to mind, just say so. So, using this finite impulse response model allows the true hemodynamic response pattern to emerge. Let's use this, this one I didn't talk about yet, but another sort of restrictive assumption has to do with parcellations, or just taking a few brain areas and doing the analysis on them. So obviously if you have a very clear hypothesis about certain brain areas, that's great. Take 3 areas, look at how they correlate, make a structural equation model, DCM, whatever you want to do. But if you really want to understand what the networks are, the whole brain, task-based networks, it's better to not parcellate, right? Just use every voxel. Because some people say, well, when you parcellate, you're reducing the noise that you have because you're taking bigger chunks of the brain. But look, I won't show it again, but the picture of the working memory, all those different networks, if you happen to include in a parcel 2 different networks, that won't decrease error, that will increase error because the signal that you wanted to retrieve is just getting averaged together into something that doesn't even exist. So it's best to use the smallest brain areas possible, so that we have that there. Each voxel can participate in multiple networks, so that's a dimensional approach that contrasts with the Yo networks where every single brain area has only one network. Impossible, right? If you really wanted to understand what the networks are that were involved in particular tasks, you'd have to let some brain areas be involved in multiple networks because even if an area, there's a lot of neurons in that area, but even if an area was partially involved in that network, partially involved in another, they might have a low weight on one network and a medium on the other network, but you just have to allow for that possibility, right? And we have timing information, that's good, and we want to extract from related to task timing. So I think I've already said all that. Okay, this is very busy. Basically what this shows, yes?
Sorry, just a question on the network definition. So how would you define a network? Is it regions that show the same true response modulated by finite?
Yeah.
How is it defined? So
okay, let's look at a picture.
How do I find the networks? So how do I know that?
Yeah.
What is the pattern recognition?
Right. So, okay. So what we do, and I have slides about this, but we may not get to them, so I'd be happy to just explain now. But so what we're doing is we're doing principal component analysis. So what gets put onto the brain, there's component loadings, right? So here I've thresholded the component loadings. How did I do that? Just chose 20%. We don't actually do any statistical analysis on the brain itself. What we do is do the analysis on the hemodynamic response shapes. So we just use repeated measures of ANOVA, and then we just use the time as one factor and the conditions of the task as another factor. And if there's a significant interaction between conditions and time, then we say they're significant. And so and this whole sort of dichotomous thresholding, yes or no, this brain area is active, starting to realize and so when we extract, say, 4 networks from an analysis, then we do have some templates of the networks that we use, the way we correlate, spatially correlate them with to try to classify them. And so that, looking at the correlation is the first step, but there are actually specific patterns in the anatomical networks that are indicative of each one of the networks. And I have them on the slide further. Maybe I'll hopefully have some time to show them. So if you actually are rigid about thresholding, you'll miss the patterns that are indicative of the network sometimes. You might have to go below, say, the 10 or 20%. But because we're not rigid about just yes or no in terms of those are active and looking more for patterns. I mean, obviously, the very low loadings are noise, and you can tell that because they just kind of look random. But when you start to look for patterns, you can have the correlations check for them, but you can physically check for the patterns as well. So anyway, I'm not sure if that answers your question. But okay, I'll skip this. But what this does, I'm showing here, we have these principles, right? Mix together the task timing related and task timing unrelated variance. So does this method actually focus on the variance that you can extract from the task timing, or does it just mix them together? So whatever's due to the task and not task is just mixed together. So I have, these are DCM, structural equation modeling, Granger causality, functional connectivity, the general linear model, the univariate analysis. These are just a few papers that have parcellations or sort of a summary of multiple tasks. Anyway, so I have the things that we talked about. Parcellate, is there an assumed matching process, or is the shape discovered? Dimensional analysis. So here, the fMRI CPCA is the software that we use and develop. And that, obviously I'm talking about these things because they do all four. But none of the other methods, except for PLS, which is also Canadian software, actually meets those four. So, any thoughts? No? Any questions about all that? I mean, it's kind of a lot to look at, but. So here, here's how we measure what the task, how the task affected the bold signal using those principles that I've been talking about. What does it tell us? That was sort of what I was talking about already, the hemodynamic response shape. So this is the process, the whole process that we would go through, which is extract the network and shapes. Sort of been talking about that. Classify, in answer to your question, I talked about how we classify the networks. And then the thing is, and the thing that is always a little bit surprising, and it was very surprising to me, which hasn't been published, we have it available publicly, but it's very, very difficult to try to determine the right way to explain the whole process, but the networks replicate over tasks. So it turns out that just like with resting state networks, there's a set of task-based networks that we can retrieve from, I would say, pretty much any task that we've tried to analyze. I know it's not accepted. It's, people think of tasks as sort of these complex things, and there's, the resting state networks separate and cause things, but when we are able to, this is sort of the COVID project we had because we couldn't use our computers to do more analyses, so we looked till our eyes burned out on our laptops on the networks we had, and we started to identify that they repeat and came up with these algorithms to classify. And so now, it's not just looking at one task, and this is, of course, the reverse inference trap. You look at one task and you think, like I did in the working memory, that this is involved in maintenance for working memory, this is involved in response, this is involved in, starting working memory trial, things like that. No, these networks are not specific to working memory or any task. They actually are general. So now the process of reverse inference becomes not such a no-no because we are finding the networks replicate and we're able to triangulate what the function might be by looking at multiple tasks. And then the last step here is, that involves interpreting the hemodynamic response shape and then correlating with behavior. I mean, you have to come up with individual differences and try to correlate in behavior, but I won't have time to talk about that. This is what I just explained about how we classify the networks. We just vectorize that and correlate with them with the templates. So here are the templates. Look at the correlations and take the top one. Sometimes they get mixed together, but typically we have good examples of the networks. This is just a picture of, there's actually 11 networks because this response network, sometimes the task is one-handed, sometimes it's two-handed, and obviously when it's a two-handed response, there's a more bilateral response network. So now the process is to try to determine what the function of these networks is by looking across multiple tasks and trying to triangulate on a function. All these words here are just changing all the time. This is kind of what our lab focuses on, is trying to determine what the function of these networks are. And it's really awkward having these words because as soon as you put the words on, people think that you're completely sloppy researcher, right? They're writing some word on there. No, it's not that. It's a lot of work that goes into determining what that word might be and completely willing to disconfirm our beliefs about this. So This is Nicole Sanford. She did her PhD dissertation on this topic, and she merged this working memory task that we've talked about already with another task that involved just listening to a definition of a function of an object or hearing a sound file play. So they were hearing a sound file play or thinking silently to themselves about this, and there was no response. So the reason it's interesting to merge that task with the working memory task is because there's silent attention to internal representations, thinking silently to oneself what a function is. There's also just listening to a sound file, which is not in the other task, and there's no response. Because the problem with the working memory task is that maintaining the letters and responding are so highly correlated that sometimes they merge together. So that's why in what I showed before on the light blue and the red and the green, the response network started too early because that's a combination of some type of visual attention network and the response network. It's supposed to start at the end of the trial, not early. So if that happens, that means there's been some, that's one way to tell there's been some merging of networks. The other way is by looking at the anatomy and the correlations and things like that. So anyway, so doing this, we get even more information that's interesting. So this is the response network. And so there's the 4 seconds between the peaks for that network. And then in the thinking task or the hearing task, there was actually a suppression of this network. So that means that when people aren't responding, that network actually has less activity during the task than before the trials started. So averaged over trials, there's a decrease. Kind of interesting, not really sure what that function of that is, but it seems to happen. And this is volitional attention to internal representation. So this was the one that I talked about that started later and has a very, very high activity for 6 letters and 4-second delay relative to 6 letters and no delay, or 4 letters and 4-second delay. But so we have hearing and generating. So generating is thinking silently to yourself. Hearing is actually hearing a sound file play. So when we merge these together, we hypothesize that the generating condition should be active for this network, but not the hearing condition. Okay? And previously it was sort of thought that working memory involved the same brain networks as hearing, right? That you were just imagining the sounds in your mind and that involved the same architecture as actually physically hearing. But it turns out that there's a much higher activity for thinking silently to oneself than actually hearing. So we can test the hypotheses this way by comparing the same networks on multiple tasks as well. This is the initiation networks, same default mode, nothing too interesting there. So. And then this is superior temporal gyrus, so a network that's involved in perceiving auditory perception. And this is very, very active in the hearing and not in any of the other conditions, not involved working memory at all. It's a bit surprising. So what I've done here is I've gone over a few of these networks already, and the rest of the slides could be either going over more networks and interpreting the hemodynamic response shapes, or I could also talk about the math behind all this for those of you that are curious about that. Or we could just talk for 10 minutes. So let me know what anyone's interests are. Maybe we could just kind of start the questions or the discussion, and because I'm not really sure. I find that I am very, very interested in all of these networks and exactly how they respond in different conditions. But I kind of feel like whenever I'm droning on about tasks and the way that the hemodynamic responses change, I don't think people are into it. But I'd love to talk about it. Have that, and then that is the end. I mean, I have some summary slides, and then I have the math. So, maybe I'll just sort of do my summary here. So what does this tell us? Macro scale bold networks and their hemodynamic response changes in response to tasking data is studied over tasks. So we can, because when we do the comparisons between people with schizophrenia and controls, between groups, we can use group as a between subjects variable in the ANOVAs on the hemodynamic response shapes. So we're actually doing all the statistical testing on the hemodynamic response shapes and not on the anatomical images. Or we can correlate with individual differences as well. So I'll just end there. Here are the people that were involved in that. Sophia is not here yet. Hi, we have an expert in fMRI CPCA right here. She did her master's partly in Vancouver over COVID with this terrible, Zoom connection, so it's the first time I've met her, but she's done a wonderful analysis of source memory task and schizophrenia. So Sophia's coming this summer to Vancouver to work more on this, so she'll be bringing a lot of knowledge back. Anyway, so what did you all want to talk about? Okay, yeah. That doesn't mean I'm getting down, though. I want some discussion.
I'll be brave.
Great.
Very interesting. Thought-provocative also. So I'll ask in the same tone. What do you think about some of the work that shows that you can predict task-related fMRI using resting state fMRI?
Okay, so the question, how can task fMRI be useful or different when we can predict task fMRI from resting state, right?
You could essentially, I think it's the Oxford group showing, it's a science paper a few years back, showing that you could predict the maps that you get from task fMRI using resting state fMRI.
Right.
Right?
Okay. So okay.
I mean, what does it tell you, or what's your thoughts about that? I mean, I have my thoughts, but
okay, I'll tell you my thoughts. Well, I have two thoughts about it. The first one is a very obvious answer, and the reason is that I didn't talk about the math, but I'll just quickly do it so I have a chance to see. I can no, just don't. Just one sec here. See, look at this. This is the dependent variable. This is just y equals y hat plus e. This is a regression equation. Dependent variable equals the predicted or the estimated scores and the error from a multivariate multiple regression. Y equals y hat plus e. We all know that, right? Right? Okay. So actually, what we're doing is a PCA on 25,000 y hats, the part that's predictable from the FIR model. That's 10%. So what's in this 90%? Anything that's not related to task timing, that's got to be largely overlapping the resting state, right? So if you're predicting task bold from rest bold, you already are 90% ahead in being successful because you probably didn't extract the task timing related variance in advance.
well, yeah, that's kind of aligns with my thought that we are mostly looking on those 90%, which is noise, right, essentially?
Well, it might not be noise. It probably is just not related to task timing, so it...
Not related to what we're looking for.
What I'm looking for, which is... But the thing is that we do a lot of cognitive operations that are for successfully doing the task that aren't related to the task timing, right? Things like, oh God, I forgot again, what are the instructions, trying to focus, whatever. Those are also in here, but they're still probably perfectly beautiful fMRI networks.
So would you consider that resting state and task-based are on the same spectrum, sort of, right? On one end, which is the task-based, you need to have a very specific task. You need to assume that you understand the hemodynamic response or to extract it in the ways that you represent it here. But you need to also assume that you understand how it works in different areas of the brain and that there are no responses of the brain that are not related to the task in a way. On the other side, okay, but on the other side you make no assumptions, I guess, and I'm not pro-task resting state fMRI, just to be clear. On the other side, you make no assumptions, which means that you also dissociate it from any function, right? That you're not able to sort of make the brain of all of your subjects do exactly the same thing at exactly the same time and measure how it responds. What's in the middle, right? How about very, very long tasks, watching movies, I don't know, looking at responses, calculating them back?
Okay, well, let's go back to the table. So it has to do with timing. Do you have timing or not? Mm-hmm. Right? It's movies. No timing.
Oh, why not?
Well, if there's timing with the movie, then you're here.
you always know, the same. Everybody watches the same movie, very long movie.
Well, but do you have numbers that you can use to extract some variance from the fMRI time series? Which could be like every time there's a car chase, every time the person is quiet, when it's quiet, right?
Every time you see hands, every time you see faces,
exactly.
every time,
So when we have timing, then these events, the more we know about when things happen, the more variance we can analyze separately from the overall bolt signal.
But getting away from the task related analysis, mathematics.
I tried to make it clear that it's not really about tasks. It's about anything that has timing, like EEG signals or anything you want. They're all very interesting. So, but it just has to do with if you have timing information, whether or not you use them. Because sometimes people have timing and they don't even use it at all. So I'm not saying that tasks are the greatest, but I am saying that if you have timing, you should use it. And the timing can be any types of events.
Agreed. And I can ask more questions, but I guess I would like other people to express their thoughts.
Any other people? I would love to talk more. We have one behind.
Could you talk a little bit more about the thinking and hearing difference? Because I didn't quite get if the hallucinations are more like the thinking task or the hearing task in their responses.
So it's actually a published paper in Schizophrenia Bulletin Open. The Open, because it couldn't get into the main Schizophrenia Bulletin, got rejected from there, and it was desk rejected from 5 journals before that. Because the conclusion is that what we did when we analyzed hallucinations, so actually the hallucinating events, right, when you did analysis of those events, we made a hypothesis that if the hallucination was longer, and this is a big discussion, but anyway, if the hallucination, there has to be shorter and longer hallucinations. And if there are, they have to show a hemodynamic response like the working memory. Right? So this is a long period of holding the information, and this is a short period. This peaks earlier than that. So for hallucinations, it should be the same thing. And so we had people pressing when they're hallucinating, and they press beginning and end. And then we had people listening to radio speech, and they press beginning and end of radio speech. And we separated that into short and long, short, medium, long. So the radio speech, you get the superior temporal gyrus network. Here, this. Beautiful response, short, medium, long, right? And her hallucinations, when they're pressing for hallucinations, you do not get any short, medium, long. So then you can say, well, people didn't do a good job of saying how long the hallucination was, right? But you also don't get any network that's not the response network. So we published a paper saying that we could not detect any hallucinations with fMRI. Yeah, that's why it got desk rejected, because everyone was like, nope, nope, nope. And in fact, it also said that the previously published papers on hallucinations were reporting the response network. Yeah. So anyway, yeah, we couldn't pick up any hallucinations with fMRI. But there's plenty of, it's a lot easier to find differences between people that are hallucinating in that past week, right? People with a propensity to hallucinate, not right in the scanner, but the brain is biased in a way that seems to be hyperactivity of this network, not for the event of the hallucination, but for people that have a tendency to hallucination, and possibly hypoactivity of a language network, which is more internal language. Like when you have to think how are these words related, or make a lexical decision, there's a network that involves Broca's and Wernicke's area, and that seems to be hypoactive, and this one is hyperactive. So there may be less, attention to internal, the internal thoughts are externalized, so that's the balance between those two networks. It's 1 o'clock, so, all right, thanks.